So we're going to talk a little bit this morning about microarray preprocessing. And a lot of folks might ask the question, why are we still talking about microarrays? And I'm going to echo Naomi's sentiment that microarrays still have a place in the world. And I say that because I hear the same thing probably you guys hear and maybe sometimes say, and certainly Naomi has heard. I've got some new samples, and I want to get everything out of them that I possibly can. So I want to use the best technology. And you ask them, so what do you want to use this best technology to do? Well, I actually want to measure the difference between this group of samples and this group of samples. So, okay, how many samples do you have? I have about 1,000. How much money do you have? Well, I have about $400. So, I'm making the numbers a little bit extreme, but the point is that sometimes microarrays are perfectly good for answering your question. They're very fast, they're relatively easy to use, and still compared to something like RNA-seq, they're cheap. And Naomi pointed out that one of the ways that we can improve power is by increasing sample size. So if you have a choice between something like RNA-seq and another technology like microarrays, that from many different, looking at them in many different ways, many different measures, are equivalent in terms of measuring gene expression, but you can do 5 times more microarrays, do the microarrays. Okay, soapbox, I'm climbing down. So I'm going to go through microarray technologies a little bit. I know for most of you this is going to be a review, but just so everybody is kind of on the same page. The reason that it's important that we talk about it, at least in very brief detail, is that the quality assessment and the microarray preprocessing that we're going to talk about later are often driven by the technology, not the other way around. So you get a new set of data, a new type of data, and you have to figure out how to deal with it. Each of the different microarray technologies has, I won't say strengths and weaknesses, but features that are different from one technology to the next. Different control different control probes, for example. Certainly different assay techniques. Different scanners to do the scanning of the microarrays to get the expression values, etc. And we're going to look a little bit at quality assessment. You guys have already done some of this in the microarray, or in Naomi's exploratory data analysis lab. So a lot of this will be sort of review. And we're going to go through microarray preprocessing. And really, microarray preprocessing or processing has 3 basic steps. Has background correction. Then there's a normalization step to try to make the data distributions somewhat similar so that we can later then do differential expression. And finally, for some array platforms, we need to summarize multiple probes into a single measure. So multiple probes measure the same transcript. So we need to have some way of putting them all together and make a single number or a set of numbers. And then the last thing I'll do is just show you a couple of bioconductor resources. The question people always ask when they ask themselves when they get a new kind of microarray data or get microarray data for the first time is, where do I find the package to deal with these data? So there are a couple of resources on the bioconductor website that can help there. So just going to go, like I said, quickly through some microarray technologies. Really, there are some, again, some common steps, but they vary in detail. There's the library preparation, and I've never done it, so it's a black box for me, but it varies from platform to platform and from sample type to sample type. Then there's hybridization, and again, there's each different microarray technology at this point often has a gadget, a little box that does the hybridization for you. You put in some parameters and it cooks overnight or something like that. So that's a variable as the library prep. Then for cDNA expression arrays, these are arrays that actually aren't used very much anymore, but the entire cDNA for a gene was printed onto the array, and you would hybridize to that. For oligo expression arrays, which are, I challenge you to find anything other than oligo expression arrays at this point in time. There are a number of different companies: Agilent, Affymetrix, Illumina, Nimblegen to a certain extent. And then I put down here other array types because I've seen probably 7 or 8 other companies along the way, seen their technologies. We've either worked with them directly or I've seen data packages to deal with them. So in your institution, you might have an array technology that I'm not going to talk about today. You can learn about it. Hybridization, this is just a reminder that these technologies all rely on a basic property of nucleic acids, that they like to find their mate. And unfortunately, this mate finding process is not perfect. Unfortunately for microarray data analysis, it's not perfect. Probably for lots of biological processes, it's great, but it's not perfect. But the point is that you're going to put a soup from your sample onto a microarray that has this sort of lawn of capture probes, and this soup up here, we made some assumptions about when we made the microarray. If we make the wrong assumptions, or we make inadequate assumptions, then we can be measuring things that are in the soup that we didn't expect to see. Or vice versa. We can have things in the soup that we think we're supposed to be measuring, but we're not. So this is the problem of the missing gene on the array. Sometimes we have genes that we totally missed, we didn't even know they existed. Sometimes we have genes or probes on the array that were supposed to work for a gene, but when you look at the data, they can't possibly be measuring anything of biological importance. So hybridization helps us, but it also hinders. The preparation of a library from mRNA is typically a reverse transcription process from cDNA, and then sometimes back to RNA. Fragment, amplify, and then I said sometimes you use the cDNA to transcribe back to RNA. When I joined my lab in 2002, I was joining a group that, along with a group at Stanford, had kind of developed microarrays, and I still had people in the lab who were using microarrays that they had actually hand printed. They had been dipping pins into DNA mixtures and then dotting them onto the microarray. So some of the data analysis and data preprocessing problems that we dealt with then, we don't necessarily have to deal with now. Because a lot of things have been standardized. This stuff all happens at gigantic factories now, as opposed to some postdocs sitting at 10 at night, putting things on slides, making mistakes, and dozing off. But the process of producing the array is still quite simple. Is still, in principle, the same. We have our clones, or our probes. We stick those onto a glass slide. These don't look like glass slides anymore, typically. We have our sample. In this case, this is a 2-color array platform where we're going to have, for example, a reference. This would be something like total human RNA from a mix of humans, for example. And then we have our sample. We're going to label one in green, one in red. Once we get it onto the array, after we hybridize it, cook it, let the nucleic acids anneal to each other. We wash things off, and then we measure in 2-color arrays with 2 different lasers a psi3 and a psi5. And then we can make this false color image where you have green, where you have essentially only reference and no sample. You have yellow, where you have a mix of both, and red, where you have vastly more sample than reference. This is what an early microarray printer looked like. As you can imagine, things like these are literally just pins. These pins would get dull over time. They would scratch the slides, things like that. So it was actually kind of fun to look at these arrays after they were done because you could see people's fingerprints. You could see things you didn't want to see. Let's put it that way. Synthetic oligonucleotide arrays are different than that. We're not typically starting with the whole transcript because it's hard to make oligos multiple kilobases long. So we're typically starting with shorter nucleotides. And these are made, in many cases these days, in silico. Or they're made, not in silico, in situ. They're made actually on the array directly, and then the array ships out to the customer. So in the case of Affymetrix and actually Nimblegen arrays, you basically start with a process, you start with a mask. And this mask blocks light from getting to the array. And you have a different mask for each of the 25 nucleotides. And in a 20, say the nucleotides are going to be 25 base pairs long. You have a different mask for each one, actually for each base for each one. And if you want to add a base, you deprotect the spot on the array. That allows this deprotection to fall off, and then you pour on the next base. That base sticks to the array, and you build things up over time, or build things up a base at a time. With Affymetrix arrays, part of the reason that Affymetrix arrays were so expensive and not very flexible was that these masks are very difficult to make. You can imagine all this is happening in a very small space. So these masks are made once say for the human genome, and then they're used many times. This company Nimblegen, which interestingly had to set up its headquarters in Iceland because of some patent issues, because the technology was potentially very similar to Affymetrix. They decided instead of using masks that were physical, they could rip the insides out of LCD projectors. Those those projectors have little micrometers in them, and by just redirecting the micrometers by computer, they could make a mask immediately, instantaneously for anything they wanted. So Nimblegen's claim to fame was you could order an array with anything you wanted on it, and they could send it to you in a few weeks. So for people who weren't working with model organisms, who had their special organism, Nimblegen was a savior at the time. Affymetrix arrays we're going to be spending some time with in the lab, and these are still the most popular arrays. I think they're about 50% of the arrays in NCBI Geo. The earlier arrays, and these are still used, so I'll talk about them, were probe sets for a transcript, and there were typically about 11 probe pairs for each transcript. They were also typically 3' biased. They were near the 3' ends of genes. And these probe pairs were designed in a very specific way. If we had a transcript, we had the sequence for a transcript, we would get these 11 probe positions, 25 mers typically. We would design those. And then in order to account for non-specific hybridization, Affymetrix built a separate set, or a related set of probes called mismatch probes. The mismatch probes and the perfect match probes were identical except for one base in the middle, one base wobble in the middle. And what they thought that this would allow them to do is to, again, help correct for these non-specific hybridizations. So when you hear me talk about perfect match and mismatch, perfect match is what we actually want to measure, and the mismatch probes are to help us, were designed to help us correct for this hybridization to other things. In practice, a lot of people ignore these mismatch probes now. And in a typical, just to give you an idea, in a typical Affymetrix array that used this kind of approach, about 1/3 of these mismatch probes actually had higher signal than their perfect match probes. So, so they weren't doing exactly what Affymetrix thought they were going to do. Yes.
Is there a reason why it was biased towards the 3'?
It had to do with the way that the library prep was done. The 3' end was relatively easier to get to, and actually in terms of degradation. So anybody who's dealt with RNA, the degradation is typically from the 5' end. So the 3' end is relatively protected from degradation. So we focus there because it's easier to get to, and also because if you have a slightly degraded sample, you're less likely to have problems associated with that. Okay. So again, coming back, this is sort of a high resolution view. Probes for older expression were at 3' end of the gene. The newer exon and whole genome arrays, or whole gene arrays, have probes that are more evenly distributed throughout the gene. And they use random priming. So rather than priming from the 3' end of the gene, which was the reason for this part of the reason for this 3' bias, they can use random priming to try to hit genes along the whole length of the gene. Yeah.
With the mismatch probes and the higher signal, was that from genetic heterogeneity in like human populations, or was that just weird hybridization?
So these are 25-mers. And unfortunately, 25-mers are not very specific. And then when you account for the fact that hybridization can happen even for nucleic acids that are quite different than the probe itself, they can still hybridize. You can get this situation where even though the mismatch probe is not actually in the genome, there's something there are many places that are very similar to it, so it can still hybridize. It turns out that this process is actually a lot more complicated than that. The base composition of these probes, and not just the base composition, but the locations along of the bases along the oligo affect both the hybridization and actually the cross hybridization. So there are methods that take into account for example, the GC content of the probe as you go along the probe and model that specifically. There's one called GCRMA. We're not going to talk about it too much, but we can talk about it in the lab if you want. Because this hybridization process is when you get back these results, it's complicated and not perfectly understood. So, Agilent, which was an offshoot from HP. How many of you own HP printers? It uses really something very similar to the HP inkjet printer technology, except instead of squirting ink, it shoots out oligonucleotides. And this technology is again kind of like Nimblegen. Now, well, at the time when Agilent started doing this, you could again send to Agilent a list of oligonucleotides that you wanted. They could print them on the array in small batches, but they could be totally customized. Another advantage of this technology over Affymetrix technology is that they can make very long oligos. So, and I believe they still do it this way. How many of you have used exon capture kits or capture kits for sequencing? Yeah. So if you've used one from Agilent, typically what they do, at least if I understand it right, is that they print a microarray, basically, with the capture probes, and then they strip them off and make your capture reagent. So they use the same technology to make their capture probes because it's so flexible. Agilent arrays. Actually, I'm not sure that they're the second largest supplier of arrays anymore. I believe it's probably Illumina. But they have typically 60 to 70 nucleotide probes. This 44, 185K, and 244K standard sizes is outdated. They're into the millions of probes now. And this brings up a point that I'm going to make in a couple slides as well. We're now to the point that we have more area, more real estate on an array than is required to measure any transcriptome. So we can now do several samples per slide, which reduces cost, or we can use multiple probes per gene or transcript. So introduces some correlation, as Naomi would say, some correlation that we could potentially either use to help us get better estimates or account for when we do our analysis. Nimble-gen arrays, we talked about them a little bit. Very flexible. When I started, we could get 2,000 spots on an array. This was great. And people who had these arrays in our lab, they were in heaven because most of the people were working on data from 5 years ago. 8,000 spots. This is the year 2000. Sorry, the years are getting cut off here. This is 36,000 spots in 2003. These were still spotted, but now by a robot. And these were the typical Nimble-gen arrays of 2004, 400,000 spots. And Illumina is up into the millions of beads at this point. So, and these are numbers really from a single array, and just for when I talk to other groups, Excel doesn't work anymore. The increased density brings along implications. You can have multiple samples per slide, or there are companies that make batch processes for these things. So, for example, Affymetrix has a system that can do 96 samples at a time in sort of a robotic fashion. We use a robot to do our Illumina arrays. We do them multiple at a time. A single sample may be on two physically distinct parts of a slide. So again, this is a bias or a covariate that we might need to account for. We can have multiple probes per transcript, again, because we have so many probes on the array. They may be identical. For Illumina and Agilent, the probes for a single transcript are typically identical. They're just multiple of them. And they may be spaced randomly across the slide. That helps us if we have a scratch in the slide, or if we, for some reason, get only half of it into the hybridization chamber, things like that happen. You're still relatively safe because you've got many copies of the probes. But they may be different also. For Affymetrix, the probes are actually different. So they're supposed to be measuring the same thing, but perhaps not. And then this leads to a real need to to understand the array platform that's being used in order to do a great analysis. You can actually do a lot with someone handing you an Excel spreadsheet and saying, here's my data. But the more that you understand about the array platform being used, the more that you can do to maximize the efficiency or the effectiveness of that array platform. So quality assessment?
So I guess,
Oh, yeah.
you know how the beads work?
Yeah, I don't know why I didn't put the slide in. So it's like magic. So Illumina has, and I've never been there, but my boss has, and Illumina has these microbeads. They're 3 microns in diameter, okay? And on each of these beads, there are, I believe the number is about 800,000 oligos. These oligos are, 50 mers or so. So all the same. So 3 micron beads, several 100,000 oligos, 50 mers, they're all the same. There's a different bead, a bead type they call it. There's a different bead type for each oligo. If you want to do the whole human transcriptome, you maybe need 36,000 different beads, 36,000 different bead types. So they create these things. They have big vats of them. And then they create a slide. And in the slide, they machine, actually I think they use something more like lithography, is used to make chips for computers and stuff. They machine in arrays of 3 micron divots that are going to match the 3 micron beads. And then they take a slurry of these beads and they dump it on the array. So now you've got these 36,000 beads, 10 million holes. They all drop in. So what do you do now? You've got these beads. You don't know which one's which, right? They just went randomly on the array. So every array that you get from Illumina actually comes with a CD or a DVD that describes what's in every one of those 10 million holes. They actually do a set of hybridizations before they send the slide to you, and they use combinatorics and barcodes to figure out which bead type is in every one of those 10 million divots. The cool thing about that is if there's a divot that doesn't have a bead type in it, that didn't get filled or it isn't understood, it just gets left out of the analysis. So, and then of course Illumina software needs to be used to translate each of these slides because they're all unique into the data that we end up using. Does that make sense to everybody? Okay. That's a good question. So quality assessment. Just a few plots here. Boxplots. You guys have seen boxplots before. But now we're going to transition a little bit to start thinking about what Naomi's going to be talking about. If we're going to be measuring something like a difference in means or so let's just talk about differences in means. Do a t-test between samples. And we start with arrays. These are box plots of the intensities from the Affymetrix microarray data that you guys are going to be working with this afternoon. If we're doing differential expression, do you think that this is something we want to start with? Look where the medians are. These things are off by quite a bit. So if we start picking sets of samples, let's say we take these 3 samples and we compare them to this set of samples. Well, we started with arrays that look different. They have different medians. An assumption that we make, and this is potentially a wrong assumption, but it's one that we typically make, is that the amount of RNA that's in a cell is somewhat constant. So if we're comparing, for example, this cell or this population of cells with this population of cells, we don't expect that the intensities, which are sort of relative measures of the amount of RNA in the cell, are going to be different, at least not their medians. Okay? That could be totally wrong. In fact, there's probably some evidence that suggests that it is wrong, but it's an assumption that we usually make with gene expression data. So we somehow need to pull these things together. So that's going to be that's one way of looking at the data. We somehow need to make the amount of RNA comparable across the arrays. Yep.
Would it be a matter of the amount of RNA in the cell being the same, or the amount of RNA that's being put onto the amount of sample that's being put onto? Because before you put it on, you can quantify it and have a notion that they're all getting the same amount, right?
So the RNA quantification, in order to put the same amount onto the slide, the assumption there is that the amount of RNA in the cells is the same, right? If we knew that the amount of RNA in the cell was different, we could actually account for that, right? we could put, for example, an array that or a cell that has twice as much RNA. We could put half as much RNA on a slide, and then all our normalization would just work because we just fix everything and make it twice. So, but it's a good point. That's what we're actually doing is trying to match the amounts of RNA. We're trying to match the amounts of RNA going on the slide throughout, because that's our assumption. It should be equal. So this is another way of looking at the data. And, so the box plots, we could see, we can see roughly the spread of the data. Here, we can see the distributions of the data. So it could be possible, for example, to have the center of the data look the same, but have the tails be quite different, or by combinations thereof. So again, one of the assumptions that we're going to have to make is about the distribution, not just the amount, but sort of the distribution of RNA or expression measures on the array. With this one, for example, you can see that the estimate of expression is actually much higher, and it's much broader. It has a broader tail. There might be a reason to believe that that's the case, but typically, again, we don't make that assumption. We make the assumption that these distributions should be somewhat similar. And by somewhat, I really mean somewhat. In some cases, we're going to assume it's identical. In some cases, we're going to assume that the variance is very similar. In some cases, actually, we're just going to assume that the mean is the same. Naomi's got a point.
I've got a question. Would you be worried about that orange one that's larger, right?
This guy?
Yeah.
We might. So this comes back to points that I brought up during the hierarchical clustering session. We can use hierarchical clustering to generate hypotheses. The same thing we could do the same thing here. We might assume that because this density looks so different, that there's something wrong with that array. We might assume that, and that's what the point I made about looking at the array platforms. A lot of array platforms have built into them specific controls. So one of the things, before we start tossing data out, we want to look and see, do we have evidence that this is not biology? Okay. Sometimes you can't tell, and you do just say to your biological collaborator, or if you're doing them yourself, you just do the array again, if you're just not sure. But this may be the one mouse out of your 6 back crosses that produced the phenotype of interest. In which case, you might not be able to remove it. You just never reproduce it. So I agree with Naomi. In all of these plots, you're looking for problems, but just because you see a problem doesn't necessarily mean that you can start willy-nilly throwing things out. When you see this, though, you do need to start digging in, and the control probes can be useful for that. Other kinds of plots can be useful. Our goal, though, is to make this look more like the other arrays. So that's going to be the rest of the talk, really, is normalization. In terms of digging into details, most of the arrays, actually all the arrays, of course, they're physically on a slide. And when the scanner, microarray scanner, gives us back data, it also tells us where on the slide the expression values came from. So we can use those expression values to make pseudo-color images of what was actually on the array. So if you do that for some of these things, you can see that yellow spot up there, that was a bubble. Happens. These are scratches on the cover slip. That's not biology. Anybody want to try to identify the person by the fingerprint? So when you see things that you don't understand, you need to start pulling in as much data as you can, as much information as you can. And Bioconductor tries with the platforms that it knows about to give you as much of that information, and often in a convenient way, so that you can do these kinds of things relatively easily. Some potential sources of variation, the amount of RNA in the samples can differ. The yield at any step actually along the way of producing the library. The label incorporation can be different, so these things get tagged with dyes or labels. The temperature of the hybridization can differ, and along with this, the humidity, etc. The RNA may be degraded in some samples more than others. And you actually see in Affymetrix, because we have probes that go from the 3' end to the 5' end, you can actually measure the RNA degradation by looking at the intensities of the probes across the transcript. So you'll hear people talk about RNA degradation plots. And we know RNA degradation occurs. The tricky part here is to know when one sample is degraded much more than the others, because that's going to affect your analysis. If they're all degraded the same amount, basically if those degradation plots are approximately parallel to each other, then everything is, quote, the same. And since Naomi's going to show us how to look at differences, if our biases are similar, then our looking for differences is potentially not so much affected. Naomi?
Yeah. So there's something in some of the software called the RNA degradation plot, and one of the questions people always ask me is, well, gee, it looks like there's terrible degradation in my sample, but really you should think of it as an average 5' or 3' plot, and just each array has a characteristic because of where the probe locations are. So on some arrays, these are very flat like that, but on some of them they do have a tilt, and that doesn't mean your sample is bad. It's a characteristic of the array. So differences are.
Yes. But similarity across the sample set is often what we're looking for. Buffers can differ, stringencies of washes, scratches, ozone we talked about yesterday. Not in here are all the HR kinds of things, the tech left, the postdoc left, etc. Other quality assessment tools, clustering we talked about a little bit. Many array platforms have specialized control probes that can be used for forensics or unusual or problematic arrays. So now we're getting into the data processing. Any questions on the QC side of things? Okay. I'm going to talk really quickly about background correction. Then we're going to spend a little bit of time on normalization and just touch on summarization. I did a quick PubMed search this morning for microarray normalization or preprocessing, and it turns out that there are 900 such publications. So I thought about just going through the first 100, but I figured it's Sunday morning, we'll skip that part. So, I'm going to summarize my sort of understanding of those 900 publications, having read maybe 10 of them, in the next 10 to 15 minutes. Background correction. What is background? Well, background is a mixture of things, but it's basically the intensities on the array that are not related to our signal. And that can happen for lots of reasons. Fingerprints, scratches, non-specific hybridization, not washing the array well enough after hybridization, cross-hybridization to specific non-targets. For example, we've had situations, and for those of you who work in cell lines, you've probably had mycoplasma contamination at some point. Mycoplasma is not supposed to be in the human genome, so that can be a hybridization, that can produce a hybridization artifact. The distribution of the background has outliers. Now, getting back to your question about hybridization and sort of effects of hybridization. The effects of hybridization differ. This background, even the background effects of hybridization differ based on the content of the probes. So if we're going to model background, some people actually model it based on not just the location on the array, but actually the content of the probes themselves. Longer oligos may help the issue, again, because the longer oligos are going to tend to have less variability in their GC content. They're also going to be more specific. Because they're longer, they can be a little bit more specific. And approaches to background correction vary quite a bit from array platform and array manufacturer to array manufacturer. So what a lot of folks do with some of the newer array platforms is they take the word of the array manufacturer. The array manufacturer is going to give you back expression values, and they may already be background corrected, or there may be a background associated with the expression value, and a lot of folks just use that. Agilent's one, Illumina, a lot of people use the Illumina software output directly. So I'm not going to spend much more time talking about it other than to know this is a step that's worth thinking about. Normalization. So after we've taken out the background, we still have expression values that may vary, expression density plots or box plots that may vary across arrays. So I'm really going to be talking about one-channel arrays here, but it applies to two-channel arrays as well, two-color arrays as well. We're trying to remove systematic biases prior to the analysis, and we want to do that while maintaining the biological signal. So we're going to assume some things, that the amount of mRNA in the cell should be similar, and we're going to assume typically that the changes in the gene expressions and gene expression measures, relative gene expression measures, and I'm going to call those M values, and we'll talk about it in a second, should really not be strongly related to the average expression. I'll give you a picture here in a minute. We're going to also make some other assumptions based on normalization methods. These others are things that we need to think about. And I'm going to show you some really strong counterexamples, or really strong, really good examples where if you make these typical assumptions and you blindly use somebody's normalization method, you can essentially eliminate all biological signal. So one thing that helps me think about normalization is the MA plot. Here's what it looks like. The M values are relative ratios. So ratios of one sample to another, or on a two-color array, the red channel to the green channel. And then along the A axis is the average expression. So you basically just take the average log expression. So log sample 1 plus log sample 2 and divided by 2. All right. Here's what it looks like in math. The two-color version, we have the log of red over green as the M value, the log ratio, and the A value is half of the expression, half of the sum of the expression values. For the one-color version, we have log of x over y, where x is one sample, and y could be another sample, or it could be a pseudo-sample. For example, we could take the median intensities from all the arrays, and that could be our pseudo-sample. And then A is just, again, the average expression. So when we look at this MA plot, what do these assumptions mean graphically? Well, to have the same amount of RNA expression, or to have the same amount of RNA in the sample, we need something like the mean or the median of the M values to be 0. So we need the center here, whatever we're going to call the center, we need that to be on the 0 line. And we also make another assumption that low expressed genes or high expressed genes, low intensity probes, high intensity probes, really shouldn't be differentially expressed based on their intensities. We don't expect, at least in most situations, we don't expect high intensity probe or high intensity genes to be more differentially expressed one way or the other than low intensity genes. So what does that mean? That means if we sort of draw a moving window or moving average through the center of this cloud of points, we'd like this to be near 0 also. Okay? So this is really the framework that I have a little mind. This is the framework that I think of when I think of normalization methods. We're shifting these points around as we do our normalization. Common normalization methods. So we talked about having similar amounts of RNA in both samples, if we're talking about 2 samples at a time. We can think about shifting this cloud up and down by one parameter, the mean or the median. We can move things up and down by the mean or median, and that gets us in the center. We could also assume that the distribution here is supposed to be similar. Remember we talked about box plots or density plots? We could assume that those box plots not only have to have the same center, but they should have the same variability, the same median, or the same standard deviation or variance. So we could also scale by the standard deviation or a robust standard deviation, the median absolute deviation. So we could shift things and we could shrink or expand things to make the distributions similar. These top ones are called parametric methods because they rely on essentially normal distributions. And then we have another set of methods called non-parametric methods. We can, for example, match the quantiles to a common distribution. I'll show you that in a second. We could use an invariant set of genes across samples. Or we could use something like a moving regression analysis. Again, for normalization, we want to shift the boxes up and down and maybe shift the boxes in size. Scale normalization, we call that, mean or median centering. How are we going to make these densities the same? Shifting the mean or median is just going to move these shapes up and down, but it's not going to change the shape, right? We could change the standard deviation. We could shrink or expand these things by essentially multiplying or dividing by the standard deviation. But what if we want to have stricter control? What if we don't even know that the shape of these things is normal? Well, we can use something called quantile normalization. And this is actually, it sounds like a big word, but it's actually pretty straightforward. You define a source distribution. The source distribution is what you're going to start with, what you're going to actually be correcting. And you need to define a reference distribution. What are you going to make your source look like? You then sort the source and the reference distributions just from lowest to highest. And then you take the highest ranking member of the source distribution and you reassign it the value from the reference distribution. And you do so for all the points on down. So what this ends up doing is all of the numbers, all of the expression values in your source and your reference will be identical while they're sorted. After we're done sorting them, or after we're done doing that, all the numbers are identical, then we put the source back in order. Okay? We put it back in the original order. So all the numbers are the same as the reference distribution, but they're back in the order that the source distribution started with. So what that ends up doing is making all of these densities identical. If we start with one reference distribution, all of these will match the reference distribution. So in doing that, everybody now has the same standard deviation, everybody now has the same mean, everybody has the same median, etc. The densities are identical. So you can imagine that's actually a pretty rough thing to do to arrays. We're saying all the arrays have to be identical. They're drawn from the same distribution. So some people have, so the underlying assumption here is that the amount of RNA is equal, yes, but the distribution has to be identical. That's not always going to be the case. So some people have some people have tried different normalization strategies that might be a little less stringent. Invariant set normalization, though, is somewhat similar. You choose a source distribution, you choose a reference distribution, and you find features, probes, that have similar ranks in the source and reference distribution. So basically, you're choosing a subset of the probes that look like they are not changing expression from the source and the reference. And you use those, basically, to do your correction. The rank invariant probes started with the same expression values, and all we're doing is correcting the rest. Okay. So in terms of an MA plot, essentially what we're going to be doing is choosing a subset of the probes here that have the same rank, that didn't change expression. And then we're going to use a moving median across here, and then we're going to subtract that out. Okay. Another technique called LOS, local regression, which is basically just a very sort of robust way of fitting a function to the points in this cloud, and then we can use that to subtract out and again shift them all toward our m value of 0. I just want you guys to know what these things are, and you're going to be applying some of them this afternoon. There are many others, and I'm not going to try to touch on them all.
The reference distribution that's generally selected is the standard normal distribution?
Good. That's a good question. The reference distribution is either, so it's a distribution drawn from the data typically. So it could be, for example, your best array, if you had a best array, or it could be the median of all the arrays, or it could be some robust choice of expression values from the arrays. But the point is, it's going to look something like these. It may actually be one of these, but it might just be a conglomeration of these. And coming back to Naomi's point, if we do this in sort of a robust way, we might want to exclude things like this or downweight things that don't look like the others. So I'm brushing aside a lot of details here. Choice of the reference distribution is an important one because that's what we're going to match to.
But it's drawn from the data.
It's drawn from the data. Typically, it's drawn from the data. Yeah. Not all arrays are gene expression arrays. Stupid thing to say, but the number of times that I've seen people apply normalization methods, normalization methods in particular, that were designed for gene expression, assuming that the samples are supposed to have the same signal, that they shouldn't be changing over intensity, is large. Array CGH, everyone familiar with array CGH copy number analysis? Okay, we're going to go through it a bit. SNP arrays, they're arrays, but we can make some assumptions, some further assumptions about how the probes on a SNP array should behave. There should be, for SNPs, they should be basically homozygous reference, heterozygous, or homozygous mutant. So those are those distributional assumptions can be quite a bit different. Methylation arrays. So those are arrays that measure methylated, unmethylated CpG sites. Again, they look quite different. I'll show you an example. Tiling arrays. I showed you an example the other day of a chi-squared distribution or chi-squared test applied to those. Different assay technologies may need very different assumptions about the underlying data behavior. And this is the flip side of this point. Not all arrays are run on arrays. People come to me with TACMAN assays, metabolomics data, many other high throughput assays that produce tables of numbers. Some of the techniques that we just talked about might actually work for those technologies. So it's worth thinking about adding these kinds of ideas to your armamentarium. We worked with Illumina early on in the development of their methylation arrays as a beta test site. So one of the things that we had to do was to come up with an approach for normalizing the data. What's plotted here are so-called beta values. The way these arrays work is pretty simple. You have a probe for each CpG site. You have an unmethylated probe that matches the unmethylated CpG, and a methylated probe that matches the methylated CpG. So when the CpG site is unmethylated, your unmethylated probe should have a very high intensity, and your methylated probe should have a very low intensity, and vice versa. If you essentially just take a ratio of the methylated to the unmethylated plus methylated, you get a distribution that looks like this. So much of the stuff, many of the CpG sites are unmethylated. Many are methylated. Very little in the middle. So if we try to make assumptions here about, for example, normality not working, right? We have values that go from 0 to 1. They're bimodal. Another interesting thing about methylation is that global methylation can change quite a bit. So you can have samples where you essentially have no methylation, samples where you have almost full methylation, and anything in between. So again, making assumptions about everybody should have the same distribution just don't work. Sven Bilke, a physicist in our lab, came up with a normalization strategy that seems to work reasonably well, at least for the early arrays. CGH is another example where our assumptions about gene expression don't work. It's pretty simple. We have tumor DNA, we're going to label it in red. Normal DNA, we're going to label in green. We're going to hybridize to now DNA. This is not RNA. This is DNA. If everything were copy number 2, if every location in the genome, in human genome, had 2 copies in both the tumor and the normal, then we would see essentially normal copy number, yellow, on the array. Where we see amplifications, I'm a cancer researcher, we might think about oncogenes, genes that are have many copies in order to overexpress themselves. DNA loss, tumor suppressor, might harbor tumor suppressor genes. When you plot it along the genome, it looks kind of like this. This is a normal chromosome 8. Down the center here is approximately 2 copies. You move over to a cancer sample. These are regions that show loss. These are regions that show gain. And so you can see that at least in this case, there's not much of the genome that's actually normal in copy number. What does this look like? Well, this is our friendly MA plot. And you can see in this case, our data are sort of centered around 0. But let's think for a minute. What happens when we have increased or decreased copy number? We said with gene expression, when we have increased intensity, we wouldn't actually expect more increased differential expression or decreased. With copy number, though, if we have loss of a place in the genome, our reference sample doesn't change. So, for example, our G value doesn't change, but our R value does. Our G value doesn't change, but our R value has to decrease in order to have a loss. Well, when our R value decreases and our G value stays the same, we also have a change in the ratio. So our signal here is all of this. What if we normalize all of this so that we try to bring all of this back to 0? Well, we just got rid of all our signal. So this is a really good example of a situation where if you apply gene expression normalization methods, you'll essentially remove a large chunk of your signal. Another example, this one you can actually see two different populations of loss, which probably represent heterozygous and homozygous loss.
For array stages, do you align them to the genome so that you can compare to the labeled regions?
Yeah. Yes. All of this is done with. So the question is, do you do this by essentially lining the probes up along the genome and looking at other probes? Yes, we do that. So anytime that you're applying a nonlinear normalization to your data, you want to think about what is it actually doing with regard to my signal? Am I removing signal that I shouldn't be removing? So summarization of multiple probes. Modern arrays often include multiple or identical probes, identical or different probes meant to measure the same molecule. And summarizing the multiple probes can potentially improve our estimates, right? We can average out some of the noise. The approaches for doing so really are going to vary, and they're going to vary based on the platform. If, for example, all the probes are identical, then taking a simple mean might be fine. The mean, though, is sensitive to outliers, so the mean might not be the right thing. We might want to use something like the median instead. We might want to use a robust mean or median, something that trims off the outliers first. And in the case of RMA, which you guys will be using for both Naomi's lab and my lab, use something called median polish. I'm not going to go through it, but it's a robust and iterative approach to summarizing probes on an array. Practical concerns. Many of these array manufacturers supply software that does the image analysis. Most of them these days come with like 100-page manuals. It's worth at least understanding the output so that you can determine how to best approach the data. This is typically going to happen for data that you haven't seen before. It's worth getting the manual and looking over it. Many array types can be handled using similar analytical tools. So if you get a new array type and you don't have somebody who's seen it before, you don't have the tools, you don't have tools that have been developed before, try the ones that you already know. And they'll often work. And some array packages deal with many types of arrays. The Limma package, for example, which you guys will be using this afternoon, can deal with lots of different types of arrays. But it does it in a very general way. There are other array type specific platforms, the Affy package that you'll be using for my lab, and the Lumi package for dealing with Illumina arrays that sometimes offer additional features that are specific to those array platforms. And lastly, just to jump over to bioconductor resources really quickly. So on the help page, there are now workflows, and there's a workflow for oligonucleotide arrays, which happens to be the top one. Thanks. And there's a little bit of a sample workflow here that has to do with Affymetrix arrays. Installation and use, exploring package content. But there's an important little piece right here called preprocessing resources. And if you scroll down through this, you can see that there are different array types. Affymetrix 3' biased arrays. That's what I was talking about mostly today. There are packages named here that work with that. Besides these Affy, GCRMA, Affy PLM, there's XPS. For exon arrays or ST arrays, you can use the Oligo package, Exon map, XPS. For DNST arrays, Oligo, XPS. For SNP arrays, the Oligo package. Illumina expression arrays, Lumi or beat array. So it's essentially a cookbook for I have a new array type, I can go here and have them lead me to the right place. So each of these has typically a very extensive vignettes. So you can actually, if there are people who are interested in doing Illumina data analysis and you want to do that this afternoon instead of my lab, go for it. You can use the Lumi package and just follow along. And I think it's section 5 of the vignette. So, all right. Any questions? Otherwise, I think some people need some coffee at some point soon. Yes.
Sometimes hear the term variant stabilization, transformation. What is that? How does it relate to this?
So when we're dealing with microarray data, if we look at something like the variance versus the intensity, what we often see, if we don't do anything special, is that the variance has this kind of behavior. Can people see that over there? Okay. The variance is high at low expressed genes, and it goes down quite a bit as we get to higher expressed genes. Variant stabilization transformations, the general concept for variant stabilization transformations is to try to level this out so that as we apply visualization techniques or others, statistical approaches that make assumptions about the variance being similar for the things that we're testing, that's actually true. So there's variant stabilization transformation, the general concept, and then there are specific applications or specific implementations of it, and the Lumi package has one called PST. So a common, it's not exactly a variant stabilization transformation, but it does kind of this thing, is the log transform. Right? If we look at the intensity, if we look at variance of the linear data versus the log data, the log data looks, the variance looks almost constant relative to the changes that are associated with the linear data. So that's part of the reason that people do log transformation, is to make these data look more normal and to stabilize the variance. Other questions? All right, cool.